become the leading biotechnology center in the United States. And we already have a good start. We have 6 academic medical centers and other research institutions like the Rockefeller University. Last year, it's my understanding that New York City gained more licensing income than any city in the country. And Seth is through the New York City Economic Development Corporation helping to build the Biotechnology Center here in New York City. He was involved in putting up a very large modern building over on the east side, not far from where I was born and raised, a couple of blocks actually. And so we're all very proud that we can be here this morning and talk about drug discovery, but I'd like to welcome Seth to give us a few words about what's going on here in New York.
Thank you very much, Howard. It's great to be here, and it's really great to see such a strong turnout on an early Monday morning. I wanted to, first of all, just welcome all of you here today on behalf of Mayor Bloomberg and Deputy Mayor Bob Steele, our Deputy Mayor of Economic Development, and to congratulate all of you on your very important work. The success that all of you have is really critical to the success of New York City, because we believe in the Bloomberg administration that in the 21st century, innovation will be the key to our remaining competitive in a very competitive global market. And the good news is that historically here in New York City, we've done very well on 2 ends of the spectrum in the field of bioscience in particular. On the one hand, we're a great headquarters city. 60%, as you know, of the largest pharmaceutical companies in the country are located in the New York metro area, and here in New York City, we're home to some of the most prolific, Bristol Myers, Pfizer, Forest Laboratories, and others. On the other end of the spectrum, we do quite well in the area of basic science, and Howard made reference to our 6 academic medical centers. We actually have 9 major academic medical centers, which is more than any other city in the world. And with our hospitals and our labs and our universities, those medical centers spin out roughly 20 to 30 businesses each year. But the problem that we faced here in New York is that we've struggled to retain these startup businesses. We certainly land some of them, but many of them end up in other parts of the metropolitan area. But we believe that we can do a better job. And so the Bloomberg administration has been working very hard to build the bioscience industry here in the city. And let me give you some examples of the work that we're doing. First of all, we've put together a series of training and networking programs. We have SBIR workshops. We have basic entrepreneurship training that literally thousands of New Yorkers have been through. We also provide access to capital. Working with the New York City Investment Fund, we've put together something that we call the BioAccelerate program, which to date has awarded about $3 million to 11 companies in just the first couple of years to help startup bioscience companies deal with the valley of death, the region between when an idea is first developed and when it's ready to be commercialized. We also have put together, working with First Smart Capital, a $22 million early stage investment fund that's available for New York City-based businesses that has also made investments in the bioscience field. And we're also working to create new space for the bioscience industry. In fact, we currently have about 1.7 million square feet of new commercial wet lab space that's under development here in New York, including the first 300,000 square feet at the Alexandria Center on the east side just north of Bellevue, which Howard alluded to, where we're hoping that the second tower will be started in the next several months. Another problem that we're working to address is something that we hear from a lot of people in the bioscience field, but in technology fields more generally, and that is a lack of engineering talent. In response to that, we issued an RFP last year where we asked universities both here in New York and around the world to propose a major expansion that they would build here in the city. And the response to this challenge was really phenomenal. In fact, in response to the last phase of this challenge, we heard from 17 different world-class institutions, and we were pleased to announce in December, at the very end of last year, that the first winner of this competition, a combination of Cornell University and the Technion Israel Institute of Technology, would be building a brand new campus on Roosevelt Island in the East River This campus is a $2 billion investment that, when fully built out, will have 2,000 graduate students, which will, when completed, nearly double the number of full-time graduate engineering students here in New York City. But we're not done even with that. And we have more announcements on the horizon. Hopefully, we will be announcing more winners in our applied sciences competition. And we also look forward to announcing the results of a year-long study called BioNYC 2020, which we'll be putting out to the public in the coming months and will provide us a blueprint for our future work. So Howard and ADDF have been great partners in everything that we're doing, and we know that the work, as I said at the beginning, that you're doing is critical to our success. So I wish you all a great day of activities, and let me just finish by saying to you that if there's anything that we at the New York City Economic Development Corporation or in the Bloomberg administration can do to help any of you with your work, I hope that you'll let us know. You can find us on the web at nycedc.com. You can call 311, or you can follow us on Facebook or on Twitter, and I assure you that if you reach out and contact us, we will get back to you right away. So thank you and best of luck.
So it really is an exciting time to be in New York. Our first chair I'm going to introduce is D. Martin Watterson, Dr. Marty Watterson, who's been a great partner to ADDF for many years now. Marty's the John Searle Endowed Chair of Professorship at Northwestern, where he runs one of the country's best academic-based drug discovery programs. And we consider Marty one of our icons of leadership in executing and delivering on drug discovery in academia. So Marty, if you'll.
Is Carl here? Okay, I was going to introduce you. Actually, I retired from running the center, and Carl was supposed to make it today. I was going to introduce you to him. Okay, so our session this morning is a watered-down version of your 8 a.m. organic chemistry class. Okay, so it's going to be oriented towards biologists, and what we're going to try to do is address some key issues that arise during the employment of the chemistry phases of drug discovery, especially the rationale. Can everyone see these from the back? Okay. So these are our educational goals here. And one thing we want to come out of this session with is familiarity that will help you with project planning, especially risk management. As you heard in the discussions yesterday, failure is a major consideration, and because of the ignorance of bliss, academics often look at the potential for success of things as a challenge. So the idea is from the session this morning and the ones following it, actually the next two following it, will expand for the biologists and those who are new to the system, how you go about anticipating some of these risks. We're going to focus on small molecules simply because of time and because there's so many precedents in that domain. So here's the simplified view of drug discovery. You'll see a lot of slides with chevrons with each stage as we expand on it, but really what you have to have is a target, which again yesterday there was discussion about single molecular targets versus pathways or functional activity screens. Then you have to have a chemical, and that chemical can be a macromolecule, a peptide, or a small molecule. And it's that interplay between the two that's critical at the early stages. So we're not talking about target validation today, but we will be mentioning the concept for pharmacological druggability. That is, when you have a target, you have something that's associated with disease progression, how do you find out if it's worth pursuing? How do you get that feasibility data? And then how do you proceed from there? And I apologize, I just checked, I noticed that there are 2 slides that are quite disturbed with the transfer between machines. To give you a bottom line, and one of these is the session focus, is that campaign risk often falls into 2 general areas, and that's adverse pharmacology or toxicology, which is historically revealed in what you'll be hearing about during the day about GLP preclinical work, and also in phase 1 clinical. And efficacy, or it's an efficacy, which is phase 2 and 3. It may disturb some people who are very proud of their animal models and the details of animal model work, but in CNS, really we don't forecast efficacy very well, if at all, in the human. So the major focus that chemists like to focus on is whether or not the target and the molecular mechanism and the biological mechanisms that they're developing their compounds for are engaged in the animal model. Are they present? And if they are, how does it read out in terms of a relevant efficacy or pharmacodynamic effect? So we aren't the only ones use this term. It was coined a number of years ago, but the idea is that the current approach is to push these failures and these parsings of decision trees down to an early stage, and this involves the use of what's often referred to as smart chemistry, and that is to make fewer, more drug-like molecules, as Dr. Lipinski has championed over the years, and then also to expand on chemotype diversity within molecular property limits. So in the 1990s, he and others have really brought this to the forefront in terms of leveraging the chances for success. One of our speakers, I don't know if you use it today, but last year you used the analogy to playing cards. Anything you can do that will pull out the bad cards from the deck and give you a higher probability of getting a winning hand is going to improve the process. So I sort of like that analogy. And as far as the biological system, although in a lot of times in biotech and academia, everybody's focused on efficacy assays, and it's nice if you can show efficacy for the same process in more than one animal model or even more than one disease model, but really it's the early non-GLP screens that address the risk. And as was referred to yesterday, this at least 2/3, if not greater, depending on what articles you read, these adverse pharmacology, toxicology issues are actually anticipated now by these GLP preclinical screens. We'll be coming back to this because now, earlier, at the very early stages of chemistry, these are now employed in non-GLP related screens. And then the other part about it is to actually early on address major barriers in the particular disease area. In the CNS, this is really a pharmacokinetic one, but this has pretty much been ameliorated over the last, I would say, 5 years plus, in terms of if possible, starting within a particular molecular property space, the probability is greatly increased for having things that start off, your hit or your lead compound is going to have some significant CNS penetrance. You're going to avoid being a substrate for unwanted enzymes such as pumps and certain genetically variable first-pass metabolism enzymes, which we'll be hearing about. A phrase often used is kill early and fast or fail fast in vivo, various versions of this. But the whole rationale here is to identify major liabilities at the start and to address at the discovery chemistry stage. So while you have the chemist engaged. And there are plenty of chemists in the room today, very outstanding chemists. So I encourage those of you who have an opportunity to sit down with them during the breaks and mixers and engage them in some conversations. This is the contemporary drug discovery engine I'm going to use as a case study to walk through what's happening currently. And I've broken it up into various modules, and the concept here is a series of go or no-go decisions. So in contrast to hypothesis-driven research in academia, you actually want to design a true scientific method approach where you want to disprove that something you have is good. And if you use standards that are in the field. There's things called SOPs, for example, standard operating procedures, which people in industry are familiar with, but in academia they're not necessarily familiar with. But those are very useful paradigms to use to parse what you're designing and making. So at the early stages, we'll be hearing more about this during the meeting, informatics-driven design, which includes structure-assisted on the single molecular target. But this can include things such as features that are correlated with a given chemotype, a given scaffold, or general molecular properties. Then there's synthesis using established chemistries. Once you're into drug discovery, you want to or pre-vet any novel chemistry you're going to make. You want to use things that are pretty standard, things that give reasonable yields, that have decent workups. And Dr. Bahana's talk, she'll be addressing the difference between some aspects of discovery chemistry, but also the difference between that and what is called production chemistry. So discovery chemistry is quite different from production chemistry, which is more of a industrial process and regulatory affairs issue. Then you have an activity module. This is concentration-dependent selectivity. This can be cell-based. It can be functional screens. There are actually ones that are in the classic mode that are in vivo. They aren't high throughput, but they depend on having a very small a number of things to test at the front end. And the key point here is the recursive nature of this. So as you're going through, you either, if it's a no, in this result here, then you have a no-go, and you either table it or kill it, or you go back to refinement. And the nice thing about this recursive aspect on all of these, where you go back to refinement, is you're rapidly generating a knowledge base in your particular campaign. So you can actually do less chemistry if you get feasibility data on the first stage. So this is also why it's very useful, and academics and biotechs can have an advantage because you have the group within limited space, or you have it very interactively. So you have multidisciplinary teams that allow you to quickly address this. And what I've put here is something that came up in discussions in a previous one, meeting, in which you actually go to non-GLP pharmacological modules. So some people like to go right to in vivo efficacy screens. If they have the model set up and their research is driven around that, they're going to do this first. But if your actual goal is to get to here and to phase 1 and 2, it can be very informative to actually do these pharmacological module screens, such as selective, pharmacokinetic, and ADMET screens. These are ones that can be miniature versions of the GLP. In fact, I list here a mini NOAL. It's no observable adverse effect level. And what you can do, this is actually an observation that's quantal. But this is actually done at the early stages of the GLP screen. It's amazing how quickly you can find out that the path you're going on, you're not going to be able to go with increasing concentrations just by observing the animals. In fact, there's a biotech company now that is based on doing and quantifying some of these clinical observations. Physical properties characterization, you want to find out early on if your computed physical properties for your scaffolds that you're working with actually correlate with experimental. Things such as computed solubility can be very far off. Things such as computed lipophilicity can be very close. And there are other ones this is controversial because some people think it's a waste of time and money to actually do some of these, but most of the efficacy models are in mice, for example, and people say it's totally useless to do any type of quick screen in mice. Well, the chemist can make tenfold less compound compared to going to a rat. It's linked, it's in the same species that you're doing the efficacy, so you can better interpret your efficacy results. Although you might go to the rat as your rodent model and your GLP. So the GLP requires a rodent and a non-rodent model. So these can be very informative, and this recursive aspect, as you'll hear about more today, helps you define where you're going to be able to go. So by the time you get down to the very expensive and drawn out, especially for chronic neurodegenerative diseases, these efficacy models, you'll be able to better interpret them. You'll be able to better interpret failures. You'll be able to better interpret the degree of success here. And then this then feeds into the actual preclinical development stage, which we'll hear more about during the meeting. So quickly, before we get into the sessions, some working definitions. Most of you know these, but pharmacokinetics effectively is what a drug what the body does to the drug. Pharmacodynamics, or PD, reflects what the drug does to the body. A key point is PD is not necessarily efficacy. Efficacy reflects a desired PD. Adverse events, which is what they're called in clinical trials, it can also be adverse pharmacology in the preclinical, reflect undesired PD, which are not necessarily toxicity in terms of these types of paradigms. This is a very important issue here, and some of the discussions that will probably come up over the next 2 days, it's good to remember this. Dosing is the pharmacological basis of therapeutics and includes the amount of the drug administered, for example, per body weight or volume, the frequency of administration, the duration of treatment, and these two are very important in terms of thinking about Alzheimer's and neurodegenerative diseases. Also, the initiation time window. Now, if the slide's not too messed up, I have one I use for teaching that has acute brain injury because that has things well defined in terms of the sequelae, but it's the same concept in terms of diseases such as Alzheimer's. And this is why you need to have some points at which you can determine initiation of therapy, for example, MCI to AD progression. And there's a lot of work going on at the animal and the clinical level to actually find those parameters that will help make that decision. The other aspect is therapeutic index. This is where I was referring to earlier about the no observable effect. You want to quickly find out with the scaffold or the hit you're working with, the lead compounds, do you have a dynamic range you can work with? Because it's a fundamental concept that eventually you will have adverse pharmacology. And the details of dosing are key in targeting widely distributed targets or complex disorders, especially if the disease state is a state of perturbed homeostasis. So this was talked about yesterday. In anti-infectives, you have a sort of a plus-minus system. And many tumor types where you have a mutation, you have a plus-minus system. But where you're actually trying to restore something towards homeostasis or delay the progression of pathology from a homeostatic state, this becomes very important. We'll be talking about molecular properties of a drug. These are physical characteristics, and Dr. Chico will address this in her lecture. And I want to use a recent and a Northwestern specific example here. I recommend reading about it. The key point here is that molecules with the appropriate molecular features can be clinically useful even if the original molecular target is not the pharmacological mode of action for disease efficacy. So one of the secrets that's kept in the closet with single molecular target work is what you actually end up with as far as the efficacy and your indication might not be the target you started off with. So this is a good case study for that. And it shows actually when the pharmacologist starts doing the dosing reaction how you can find out you're not working with that particular molecular target. So we're also going to hear about natural products in the last lecture. And it's nice to remember a few things about natural products. I'm using caffeine here. It's all familiar to us. It has two pharmacological actions. One is a nervous system stimulant, and the other is a diuretic. So this is a single drug with two in vivo pharmacological actions. So Mother Nature has already done multi-target drugs. It's also a very safe one, and this is true of a lot of natural products. It's quite difficult to actually overdose on caffeine. However, the caveat here is that natural products are not inherently safer, although marketing would have us believe everything that's natural is safe. And just remember that arsenic is a natural product. And I've already mentioned the fact that many of these are multi-target drug candidates. So actually, the organic chemist has to come up with ways of making feasible, tractable synthetic methods to start pulling away those multi-target activities if it's desired. Just a quick bottom line here, this will be covered more later, is that alternative use and reformulation of existing drugs can actually involve a lot of risk. Also, it's important to remember, just like natural products, just because it's FDA approved does not mean it's safe. You also have the risk of failed clinical trials can actually damage a field potentially. So it's always good to be alert to this because conceptually, if you're going to test an existing drug in a particular indication, such as CNS, which is the focus of this meeting, it's important to ask, you might have the target in the brain, But if the drug cannot accumulate appreciably, distribute within the brain very well, then PK is going to kill your efficacy. So interpreting that trial or designing that trial in that regard is very important. We'll also hear a little bit about the chemistry. We go to Mother Nature for some of our chemotypes. So chemotypes, the idea is these are the small heteroaromatic cores that are at the heart of a lot of drugs, and then we expand on those to get our scaffolds. And these are all ones that are amenable to chemical diversifications using established synthetic schemes, and Dr. Bahana will give you a quick overview of this concept of chemical diversification, the handles you want to. If you view these as the core Lego piece, you have to have ways to snap on the other pieces, and that's basically what you have to address at the very front end of your chemistry. So chemical diversification can either yield major changes in PD with minor chemical changes. I'll show you an example in a moment. I'm not sure if Heather's going to use the same example, but the idea of putting methyl groups and moving them around a ring, or small changes in PD, but you can actually improve pharmacokinetics. So later on, we'll hear some talks later on in the day that talk about what you do in terms of refinement from hit to lead. So you'll actually have to improve the pharmacokinetics, and depending on your scaffold and your chemotype, that may be very facile in terms of the structure activity landscape, or maybe a challenge. And these are just two examples from the literature. And this is mostly extracted. I don't have the acknowledgement on here on Camille Vermouth's textbook, The Practice of Medicinal Chemistry. But you can have a steep landscape, and this is showing you two drugs from literature in which the case of where the same scaffold, but the methyl group is just simply moved, and you actually get a very different pharmacology involved. Whereas you can have things where you have a flatter landscape where the presence or absence of a methyl group has very little effect in terms of the pharmacodynamics or efficacy or target engagement. So what does this mean? It's a wordy slide. So the take-home lesson here is that you want to find out and talk to your chemist about, does your scaffold, your hit, have a flat structure activity landscape, or is it a steep one? And there are certain implications to this in terms of what you might put into your libraries for your functional screens. It also might suggest that early on you want to work with an additional chemotype, put a little bit of effort into that. Whereas if you have one, oftentimes ones that are in this category are things called privileged chemotypes, such as if you move the methyl group around like the one I showed you, you'll actually change the pharmacology. In that case, you might want to do different early. So that pharmacology module I mentioned earlier, you might actually want to give a little bit of emphasis to saying, gee, do I generate first-pass metabolites that have quite different pharmacology so I can anticipate different pharmacologies or even toxicities. So Dr. Bahana will summarize what one looks for in the chemistry toolbox and how one generates this. And I want to end with this one because this is coming back to the concept of the interface of PK and PD and the planning issue, especially if disease progression modification is a goal. This is important because there are certain dogmas that evolve that come from a person's life experiences, not necessarily from the data. And when one is working with what are called palliative drug discovery, the presence of the drug continuously, I'm trying to think of non-specialized terms to use for this, but you actually may have to have the target engaged for a prolonged period of time to get the desired efficacy. But if you're affecting, for example, the biosynthetic process, or you're affecting something in which the outcomes, let's say the neurologic outcomes or the efficacy is downstream from where you're targeting it, the therapeutic time window is critical, and in fact, you don't necessarily have to have the drug at high levels and distributed throughout the whole body. In fact, by not having it that way, you could potentially actually decrease adverse pharmacology because you're not having the drug. So in the case of CNS, those of you who know about tricyclic antidepressants, you know about how these depot, they're very lipophilic, and that actually impacts dosing, and it also impacts adverse pharmacology. So in this case, you have this acute case, but it's the same paradigm can be done in terms of a slowly progressing disease, but you have the injury, then you have a delay that's a clinical consideration. So you may want to treat something right here, but if it's stroke or if it's closed head traumatic brain injury, this is not going to help you very much. You want to look in here for your biochemical changes that might be occurring. And that might be your biological mechanism of action, which would also possibly include a molecular target mechanism of action. And the question really becomes if you dose in here, and it could be you can go up and keep this present. It could be the compound could be intervening here and then being eliminated. But you want to actually look to see if you're attenuating your mechanism of action here, and then go further out to see if your efficacy. So this is an early pharmacodynamic effect, but it's not what you're looking for. Your pharmacodynamic effect is what you're trying to induce in order to alter the efficacy out here. So the interface of these and planning for this early on, what your chemistry colleague does in terms of the hit to lead and lead to candidate refinement become very important in this in terms of project planning. So in summary, smart chemistry is recursive and it's driven by the biological goal. It's very different from organic chemistry applied to interesting biological problems. There's a requirement for an integration of decision filters based upon informatics and feasibility screens. I gave you an example from one type of discovery engine that's a pretty common paradigm used. And then in terms of the themes and caveats, the process is similar among small molecule chemistry with details defined by a particular campaign. And this will come out in terms of the themes that are covered in the next 3 talks. So our first speaker is going to be Dr. Laura Chico, who's going to be talking about designing small molecules, increased potential for CNS bioavailability. Laura is what I would call an informaticist, especially in terms of pharmacology. She has a master's from School of Engineering and Informatics and a PhD in pharmacological sciences, and has been actively involved in academic-based drug discovery and in biotech-related drug discovery, and has been key player in campaigns that have brought, with high efficiency and rapid speed, drugs that are now doing very well in terms of the clinical trials. Heather will be talking about synthetic chemistry essentials for biologists. Heather has a training in organic chemistry, postdoctoral experience in drug discovery, and was a senior chemist at Astellas Pharmaceuticals, and is now doing work in terms of evaluation of the industry at JMP Securities. I always get the initials wrong. Frank has had a great tradition in terms of natural products and has industry experience and will be addressing the challenges and power of using natural products as a starting point in drug discovery. So Laura? And I'm purposely waiting for specific questions and answers after these 3 talks, and then we can then have a generalized open one too.
Good morning, everyone. Can you hear me okay? Okay. So, as Marty mentioned, my talk today is about designing small molecules with increased potential for CNS bioavailability. And if bioavailability isn't at the front of your mind when you're early in your discovery process, I hope after today's talk that it will be. So just a brief overview about what we will be talking about today. As some background, I'll address why bioavailability is so critical to address at the front end in a CNS drug discovery campaign. And we're going to just talk about this in general terms. So when I mention bioavailability, we're simply talking about the amount of drug that's available in the body that's available to act at a target. And in this case, these are going to be CNS targets. And bioavailability is influenced by ADME properties. You might have heard that acronym a lot already. Absorption, distribution, metabolism, and excretion. And I'll give a couple examples where we can see how bioavailability and even metabolism can have dramatic effects on efficacy and safety. And this just kind of addresses why it is so important to deal with it on the front end. And Marty kind of mentioned my talk is a risk management talk, so I don't like that term that much though. So I prefer the casino analogy. essentially, molecular properties are what's influencing bioavailability. So if you can find a way to bias your efforts, you can help kind of increase your probability of success. Now, this isn't going to guarantee you're going to find a drug, but hopefully it helps even the odds out a little bit. So we're going to be talking very briefly about specific molecular properties that you should be considering in your design efforts and how you can go about measuring those. And essentially, when we're saying molecular properties, these are just the features of the drug that make it act like a drug in the body. And again, the take-home message here is by incorporating these guidelines, you can help prioritize your efforts a little more efficiently. And so then to conclude, we'll kind of apply these concepts or see how they've been applied in the past and how they can help you in your own efforts. So to start off with, I think it's important to highlight that when you're attempting to identify CNS drugs, you have a unique set of considerations that you have to address going beyond efficacy. And quite simply, that is the challenge of getting the drug into the brain rather. As I mentioned, bioavailability, we're just talking about the drug that is available in the body to act at the target. Now, the inability to reach the brain in sufficient amounts during an appropriate, therapeutically relevant time window is what's limiting your opportunity for efficacy. And so some factors that are playing into that are the blood-brain barrier, getting across that, first-pass metabolism when you're thinking about an oral drug, and even efflux. So you have transporters within the brain pumping things back out if you're fortunate to get it to penetrate in. So I should caveat this and say that just because you have something that is bioavailable, it's not going to guarantee drug efficacy. But I think it's pretty obvious that if you don't have sufficient bioavailability during that critical time point, you're going to severely limit your opportunity for success. So just to get us all on the same page, how does an oral drug actually get into the CNS? We have a little schematic here. Upon administration, you have the drug entering the GI tract where it gets absorbed into the